Hello, my name is Amy Nguyen, and my Talaria project was on primer design for Hox genes in SMED asexual reproduction and regeneration in SMED Mediterranean through qPCR. My mentors were Dr. Christopher Arnold and graduate student Shrestha Ghosh from West Virginia University. So, to first get in some background about what my project was about, we'll first talk about what regeneration biology is. Regeneration biology is an increasing innovative field that studies the restoration or replacement of damaged or missing cells. Some of the potential applications from this research that they're hoping to find solutions to is wound healing, nerve repair, organ replacement, and correction of genetic defects. If these potential applications are able to be applied. Many of these applications would be applied to medical aspects in terms of creating more organ transplants because there's a known deficit of that, as well as, oh wait, no. Hello, my name is Amy Nguyen, and my Talaria project was on the primer design for Hox genes in SMED asexual reproduction and regeneration in SMEDIA Mediterranean through qPCR. And my mentors were Dr. Christopher Arnold and Shrestha Ghosh from West Virginia University. So to have a little bit of background about my project, we'll first go into about what regeneration biology is. So regeneration biology is an increasing innovative field that studies the restoration or replacement of damaged or missing cells. And some hopeful potential applications of regeneration biology are wound healing, nerve repair, organ replacement, and correction of genetic defects. And a lot of hopes for these applications would be to create more organ replacements because there is a lack of those, and as well as healing genetic defects such as cleft lip palates. Planarians, also known as flatworms, are common specimen models to use for studies on regeneration due to their unique regeneration. Wait, I spelled this wrong. Okay, wait. Hello, my name is Amy Nguyen, and my Talaria project was on primer design for Hox genes in Smidia, um, Mediterranean for its. Okay. Hello, my name is Amy Nguyen, and my Talaria project was on primer design for Hox genes in SMED asexual reproduction and regeneration in Smidia Mediterranean through qPCR. And my mentors were Dr. Christopher Arnold and graduate student Shrestha Ghosh at West Virginia University. So first we'll get into a little bit of background on my project. So we'll get into what is regeneration biology. So essentially regeneration biology is an increasing innovative field that studies the restoration or replacement of damaged or missing cells. And some hopeful potential applications of this field is wound healing, nerve repair, organ replacement, and correction of genetic defects. Um, those who study this field hope that it will have successful outcomes in and applications in medical fields, such as creating more organ donors, and as well as being able to correct congenital defects such as cleft lip palates. Planarians, also known as flatworms, are common specimen models to use for studies on regeneration due to their unique regeneration capabilities. And then as well, in order to have some more background about my project, I'll also explain what Hox genes are. So Hox genes are a group of highly conserved genes that are essential to animal development and embryogenesis, specifically body segmentation. So how the parts of the body in a animal develop while in an embryo. Hox genes can be found in many species, including planarians, but there are a wide array of species that have Hox genes. And what makes Hox genes significant is that if Hox genes are disrupted, it can lead to abnormalities in body patterning and segmentation, such as the congenital defects that I talked about before. So, the significance of my project in terms of the two topics that I shared before is that previous studies show that Hox genes are essential to body planning in many organisms, like I stated before. And for a long time, this was most of the knowledge about what Hox genes were. But my mentor, Dr. Arnold, discovered within the last few years that Hox genes also play a role in asexual reproduction, as well as the possibility of its role in regeneration. My project in specific aims to examine the effect of BMI1, which is a Hox gene, BMI1's knockdown on the expression of other select Hox genes in Smidia mediterranea. And essentially, the question that we're trying to ask is, does BMI1 knockdown, which is the temporary inactivation of the Hox gene, if that works the same in planaria as it does with other species and animals? So, in terms of the methodology of my project, we first did some general data information collection from a publicly available database called Planosphere. And for each Hox gene, information that was collected included SMED identifiers, genomic sequences, and other annotated features such as exon intron sequences. After that, we prepared the genomic sequences. We used the exon intron sequences that we got from Planosphere of each Hox gene, and we uploaded it to a free software called the APEA plasmid editing software. And after that, we artificially spliced out the introns, which mimics the mRNA following a DNase treatment that would usually be done in an experiment like this. After that, we aligned those sequences with other reference plasmids for ideal primer placement. So the exon only sequences after the introns were spliced out were aligned with their corresponding reference and plasmid files in order to identify the mutated and plasmid inserted regions to avoid if possible, but for some of them we weren't able to avoid them, which was okay. After that, we did the primer design and selection. Primers were designed using a platform called Integrated DNA Technologies Primer Quest tool, which allowed us to submit different specifications that we needed for the primers, which included an exon-exon junction overlap and a high amount of sequence in the upstream. In order to find primers that would be suitable for those different requirements. And after that, primers were analyzed for best efficacy and they were selected. After that, validation and qPCR procedure occurred. So primer testing and quantitative PCR was performed by my mentor, Shrestha Ghosh, since this is a process that would have to happen in person and this is an online program. But primers were tested for specificity and efficiency of genomic sequence amplification, and our qPCR was run using cybergreen dye, which is an indicator used to assess amplification of primer performance. So moving on to our results after the qPCR, we'll first analyze the amplification plot curves. So amplification plot curves show the fluorescence signal emitted from cybergreen dye over the course of the PCR cycle, which is basically during the qPCR, the selected genomic sequence that we're targeting is amplified until saturation is reached. So you can see to the right where all of the amplification plots, there is a increase where the saturation is reached. And the faster saturation is reached, the more DNA that was present in the sample. As you can see to the right, the results confirm that all chosen primers amplified their target genomic sequences, confirming their functionality. Moving on to the melt curve plots. Melt curve plots show the melting temperature of the amplified DNA products using cybergreen to determine amplification specificity. So when the melting temperature, when it denatures the product. So in terms of analyzing these melt curve plots, one distinct peak means that a single specific DNA product was amplified, which is what we want, but multiple peaks show non-specific amplification, or it's also called primer dimers. And our results confirm that all chosen primers amplified a single specific product, confirming their specificity. Next, we'll move on to the relative quantification plots. So relative quantification plots show the relative difference in gene expression between a test sample and a reference control sample. For this specific project or study, we used a negative control, the UncRNAi, RNAi standing for RNA interference, and versus the post-to-be treated worms, post-to-be as in another Hox gene that the worms were treated with. And our results to the right show that they revealed no significant differences, which means the p-value is less than 0.05 in expression in any gene between post-2B RNAi and the control worms. I've also included a reference for statistical analysis of these plots on the bottom, showing that overlap means that the standard deviation means that the data was not statistically significant, but no overlap means that there was a statistically different significance to that. Lastly, we'll move on to the Welch t-test. So the Welch t-test shows whether the means of two independent groups have a statistically significant difference. For the study specifically, we found no significant difference when comparing the mean expression of any Hox gene in control versus the BMI1 RNAi worms. However, we did find that there were nearly significant changes in three of the Hox genes. So first, we found that there was a nearly significant increase in HOX1. In LOX5B, we found a nearly significant decrease, and in post-2B, we also found a nearly significant decrease. I've included some of the relevant data from the t-test on the right, including the Hox gene, the mean fold change, p-value, and r-squared value of each Hox gene. So next we have our discussion and conclusion. So the results from this project showed that the BMI1 knockdown, which again is the temporary inactivation of the Hox gene, does not cause large changes in Hox gene expression in Schmidtia mediterranea under the tested conditions. However, there were near significant differences in three Hox genes, HOX1, LOX5B, and POST2B. After some analysis of these results, so we found a possible reasoning for the results, which is that there may be interactions between different cells in the planaria that were not controlled during the test and need to be isolated. So, in terms of what we mean by interaction of different cells in planaria, we mean that it could possibly mean that the increase of one cell can cause the decrease of another. And if that were true, that could give some sense to the results, and that BMI1 could be repressing HOX1, while HOX1 represses both LOX5B and POST2B. And if BMI1 were knocked down, like in our experiment, HOX1 would increase and LOX5B and POST2B would decrease, which is what we found in the previous results. But after that, we concluded that in order to confirm this hypothesis, further testing is needed, and we suggest that further testing should include larger samples in order to include more data and make a more accurate result, and also isolating the cerebral area of planaria of the Schmidtia mediterranea, where HOX1, LOX5B, and POST2B are found in Schmidtia mediterranea. Finally, to talk about my Talaria experience. So for any new Talaria participants, I want to share my experience to share what happened. But initially, at the beginning of this experience, I wasn't sure exactly what I wanted to do for my research project. I was passionate and still am passionate about multiple things, but Dr. Christopher Arnold was very accommodating in that fact and wanted to learn more about my general interest to hopefully focus on a specific area that would be good for the project. And in the end, we ended up choosing a project that involved working with what his lab was already working on, which is the Hox genes in planaria and also the regeneration and asexual reproduction. I also was very lucky to work with graduate student Shrestha Ghosh, who was a graduate student working in Dr. Arnold's lab, and she set aside a lot of time to help me develop fundamental basics of the project, going through very specific steps one by one in order to show me how I would be able to design the primers, since this is something that I didn't learn before, and also to go through the different databases and other materials that I could use online to design the primers. I also, both Dr. Arnold and Shrestha Ghosh did multiple check-ins with me throughout the program, answering general questions and also re-explaining concepts and tests that were essential to the project in order to have a really good understanding of the um, essential components. But overall, my experience with Talaria was very fun and rewarding. I'm so glad that I've been able to participate in this experience, and I've met so many new people, including professionals and other students through my pod in Talaria, and seeing what other people are interested in, and also getting feedback and getting new connections with other people. And it's also confirmed my interest to continue to pursue research in the future. So if anyone who's watching this is considering on applying to Talaria and being a participant of the program, I would highly suggest it because I had a very fun time. Thank you for listening to my presentation, and I hope you enjoyed. Thank you.